Hello, I'm David Kerr, Professor of Cancer Medicine at the University of Oxford. What I'd like to do today is to talk a little about a phase 1 trial that's just been published in the May edition of Lancet Oncology. A beautiful paper by Emil Liu and colleagues in which they looked at an extraordinarily complicated regime of gene editing and then reinfusing tumor-infiltrating lymphocytes. Again, just shows how remarkable modern cancer medicine can be. So I'd like to talk a little about the procedures involved, given the relative complexity. So initially, patients in the phase 1 trial with metastatic advanced gastrointestinal cancer, the first step was to receive non-myeloablative lymphocyte depleting chemotherapy. This is cyclophosphamide and fludarabine. This is followed then by high-dose interleukin-2. The process of producing the gene-edited tumor-infiltrating lymphocytes is absolutely fascinating. So the gene-editing target, the gene that we wanted to knock down, was a novel internal immune checkpoint protein, a gene CISH, which encoded a cytokine-inducible SH2 domain-containing protein. So an internal immune checkpoint target, quite an interestingly novel one. So that's a target to knock down. But the process of building the therapy is completely fascinating. So autologous tumor-infiltrating lymphocytes, TILs, were generated from a tumor biopsy, fragments from each individual patient. Samples of the TILs were then cultured with patient-derived lymphoblastic cell lines or monocyte-derived dendritic cells, loaded with pools of synthetic 25-mer peptides containing tumor-specific mutations that were detected from whole exome sequencing from the patient's tumor. So think about that for a second. So the patient undergo a biopsy, the tumor-infiltrating lymphocytes are taken off for culture, more of that later, but they were co-cultured with a source of patient-specific tumor-associated antigens, which were generated by sequencing the tumor, whole exome sequencing, and then using 25-mer fragments of what they felt were the key tumor-specific antigens, were plumbed into, the patient's autologous, their own cells, dendritic cells, so that these antigens would be presented to, at the time of co-culture, with the patient's own tumor-infiltrating lymphocytes. It boggles the mind. So the tumor-infiltrating lymphocytes with demonstrated reactivity to the neoantigens in this co-culture experiment were selected, and they were then subjected to the CRISPR gene editing in vitro. So they knocked down CISH, this the target that they were aiming for. Then the edited TILs underwent a rapid expansion protocol. They were cryopreserved, and then following some very sophisticated molecular quality control, infused back into the patients. Goodness gracious. I mean, extraordinary when you think about it. So it was a phase 1 trial, so dose escalating the number of cells. It's pretty well tolerated. Some, as you would expect, some fatigue, some fever. Nobody died in the back of any of the treatment that was given in that way. It was moderately well tolerated. In terms of effectiveness, these were sort of patients with advanced disease who'd undergone multiple previous lines of treatment. There were no severe cytokine release syndromes, nothing of grade 3 or worse, no neurotoxicity. 6 of 12 patients had stable disease by day 28. 4, that'd be 33%, had stable disease ongoing at 56 days, and 1 young adult who had microsatellite unstable mismatch repair and deficient MSI high tumors. Therefore, they've already got an existing high neoantigen load, um, had a complete response. So that was very, very pleasing. So I say again, an extraordinary piece of work to think about the complexity involved in every step of that process before the patient's own cells, the autologous cells, thus manipulated, were reinfused with moderately acceptable toxicity, I would say. And, it's a phase 1 trial, so you're not really looking for a big efficacy readout. But the one younger patient who had if you like, a genetic predisposition to responding to immunotherapy anyway, had a complete response. I'd be really interested in what you thought about it. Who knows what the cost of that would be in terms of the complexity, I keep using that word, don't I, of every single step. But modern cancer medicine, don't you love it? Don't you love it? So well done to the team for producing this phase 1 trial result. How generalizable it will be remains to be seen, given the multiple different steps that are required. But it just shows you where, in my lifetime as a cancer doctor, 4 decades, as I've said many times before, how remarkable progress has been. Very interested in any comments you'd have to make. But again, as always, thanks for listening. And for the time being, my escapers, ahoy and over and out. Thank you.